Welcome back, guys. Yesterday I wasn't able to take classes because I was a bit busy. So, apologies for that. So, let's just start before wasting any time because I don't really have much time and I want to complete this chapter today. So, yesterday we did chose cytoplasm and now we were left with the lysosomes and some of the components that I didn't mention here, but I'll be just briefly explaining them because they are not really in they're not really required in A-level syllabus. It is my mute. Okay. So they're not really required in the A-level syllabus. What we were left with, it was cell membrane, which I said that it is going to be the next chapter in which I explain cell membranes and the processes that it goes through it, the importance of it, the structure of it. Anyways, we were left with lysosomes, cell wall, and you can say some of the other components. So let's just go with the cell wall. So starting with the cell wall. So this is the structure of cell wall here. So cell wall is made up of, this is middle lamella. So this is middle lamella, this is primary cell wall, and this is plasma membrane. It is cellulose microfibrils, this is hemicellulose, and these are pectins which are connected. As I don't need to explain much because it's already here. What I need to do is explain you the importance of it. So the primary structure of cell wall is made up of the cellulose fibrils running through the matrix. So this is the matrix. This part is the matrix. And the cell wall has high tensile strength. It is very strong, and why it is it should be very strong, I'm going to explain you in a bit. But the matrix consists of polysaccharides, which are usually divided for convenience into hemicellulose and pectins. Pectins are, now this, you should listen this carefully. Pectins are acidic and have relatively high solubility. The middle lamella is the top that holds neighboring cell walls together. It is composed of sticky gel like magnesium and calcium, which are called calcium salts, which are called pectates. So, for example, this is the cell wall here. Yeah. So the part which connects or touches with the adjacent cell wall is gonna be the middle lamella because it is on the top. So this blue part is the middle lamella, which is touching, or you can say connected, with other adjacent cell walls. So now it is made up of gel-like structure, which is sticky gel-like structure, usually of magnesium and calcium salts, and they are called pectates. The other part, which is the fibrils, and you can say which is present in the matrix, which is hemicellulose, are a mixed group of alkali soluble polysaccharides like cellulose. They are made from chain-like molecules, but the chains are less organized and shorter and more branched. So cell wall is 60-70% mass of its water. Yeah. So what you need to understand here is that I told you in the starting that there are two types of organisms. One is prokaryotic and another one is eukaryotic. And the prokaryotic one's cell wall was made up of peptidoglycan, or you can say murine, yeah. So it was made up of peptidoglycan, and this one is made up of cellulose, hemicellulose, whatever you want to say it. But the thing is, as we have discussed in the chapter of biological molecules, how cellulose is made, it is made from, so cell wall provides high tensile strength. So it is made up of cellulose is made of beta glucose. Now, the peptidoglycan is not made from glucose, but it is made from a derivative, nitrogenous derivative of glucose, or amine derivative of glucose, which is N-acetyl glucosamine. You don't need to remember the structure here. You don't need to memorize the formula or the atoms involved in it, but what you need to know is the name. That peptidoglycan is made up of N-acetyl glucosamine, which is a amino derivative, or you can say nitrogen derivative of glucose. As you can guess from the name, N-acetyl glucosamine. Now, one thing more here is that there is something called plasmodesmata present between the cell walls. So for example, this is a plant cell here. It is not usually that square. I should just draw it by rough hand. So this is cell number 1, this is cell number 2, and for example this is cell number 3. So they're interconnected with each other by, yes, you can say the name, middle lamella. So they're connected with middle lamella, and so this feature is not inside the human cell because they don't contain cell wall. So for example, this is the cell wall here. Now what usually happens here is there are certain gaps which interconnect the cytoplasm, or which connects the cytoplasm between the cells. So what happens here is that nutrients, ATPs, and different kind of substances can be transferred from one cell to another plant cell, not animal cell. Animal cell cannot transfer this like this. They have to cross membrane first. But without crossing any membrane, nutrients and ATPs, for example, let's say, can transfer from one cell to another without crossing any membrane or any cell wall, just by this opening, which we called plasmodesmata. So plasmodesmata are living connections between neighboring plant cells, which runs through very fine pores in the cell walls. The cell surface membrane of neighboring cells are continuous and line the pores of running through the center of each pores in smooth endoplasmic reticulum. Communication and coordination between cell wall is therefore made easier, since molecules does not have to cross cell membrane. So if, for example, if this cell A wants to communicate with cell B, it wants a certain, you can say, enzyme, and it is lacking in cell A, and cell B have it or produces it. So what's going to happen is this molecule, which is a messenger molecule, goes in from A to C to B, or you can say directly it can go from A to B via these connections of plasmodesmata. So this molecule goes very easily from via this path without need of crossing any cell membrane, which increases the speed. So the coordination is going to be a bit more accurate, and the communication between cells is going to be more reliable and more easier. Movement is, however, regulated. Now this is the thing. It is not like that a nucleus, for example. By the way, the pores are very small. I have built relatively very large pores here, but it's not like cytoplasm is just floating here and there. As I just explained in the last class about cytoskeleton, which is present, cytoskeleton holds everything inside the cell and regulates it. So if something is to be crossed via plasmodesmata, It is when the plants need it, or it is regulated, you can say. But there is one disadvantage of this. Virus can take, you can say good things can cross from A to B very easily, but so does the bad things. So if a virus is present inside a cell A, it can easily cross cell B and spread through plasma-disc matter. So plasma-disc matter provides large molecules like ATP to transport freely from one cell to another, so does the viruses. So it's very easy for viruses to cross across the plasma-disc matter from one cell to another without just crossing membrane. So this is the thing. So at some point it has advantages, very much advantages, but some disadvantages are also attached to it. Now, coming to the this is in the cell wall of plants, as it is okay, hold on, this is very obvious. Anyways, let's just talk about the function of cell walls, and we're done with the cell wall part. So functions of cell wall are mechanical strength and skeletal structural support. Gives the ability of turgidity, slash resistance from turgor pressure, resisting cell from bursting. Orientation of cellulose microfibrils limits and helps to, and the interconnected between plasma-disc matter by middle lamella when the cell walls are connected with each other, providing a route from one to another. Cell wall have also different kind of functions, which are included in another chapter, which is basically the transport in plants. So there you will learn that how cell wall helps in the apoplastic pathway, which is absorbing water. So water moves faster from cell wall to cell wall to cell wall to cell wall, instead of going into cell, then membrane, then another cell, then another membrane, and so on. So from cell wall to cell wall, it's a bit easier because cell wall is usually like a sponge. It absorbs water. Now, let me just explain you what turgor pressure means and what turgidity or resisting turgidity means. For example, let me just draw something here. For example, this is a animal cell, and this is a plant cell here. I've made a rough drawing, so please don't come and say in the comments that my drawing is very bad, though it is. Thank you for your criticism. But anyways, so this is an animal cell. And this is a plant cell. This is cell membrane, or you can say plasma membrane, and this is cell wall, then this is cell membrane. Now, they both are placed in the water, or you can say distilled water here. So now there's a concept of water potential here, yeah? So let me just give you a overview of it. I know it's a bit hard to understand, but there's a dedicated chapter explaining those concepts. So we're going to be covering it in the chapter of again, the transport in plants. But let me just give you a brief overview or a simplified explanation of it. So the water potential is the purity of water, yeah? You can say that, although it means that how much water molecules are present with the, you can say, in comparison with other molecules. So for example, this is a glass of water, yeah? And this is distilled water, pure water. And this is a glass of water. They both are 1 liter, but this one is pure and this one contains 1, or you can say 10 grams of sugar. So the water is saturated with sugar, so it will have lower water potential. And this one, due to being unsaturated relative to this one, it will be called higher water potential. Now, higher water potential, or you can say pure water, is represented by this symbol, which is equals to 0. So if the highest water potential is denoted with 0, and the lower the water potential goes, it goes into -1, -2, and so on. So because of pure water being at 0 water potential, and the water potential inside the cells, because of the presence of components, is in negative, the water always move via osmosis, which is like diffusion from higher water potential to lower water potential. So from higher water potential, which is 0, to lower water potential, which is -10, for example, let's say, the water will move via a partially permeable membrane. This is called osmosis. So the definition of osmosis is that the movement of water molecules from high water potential to low water potential via a partially permeable membrane. So in both cases, in animal cell and plant cell, the water is going to move inside the cells. Now, after some time, when the water is full, it will now start exerting the turgor pressure or water pressure on the walls of the cells. In this case, the last wall is going to be cell wall, and in this case it's going to be membrane. So after some time, because it's not really hard, the cell membrane is not really hard, what's going to happen is, with the animal cell, after a long time, when there is a lot of water being accumulated inside the cell, and due to high turgor pressure, the cell wall will burst, killing the cell, because all the components are gonna escape, and the rest is history. Anyways, the cell is gonna die. What happens in this case is, due to the high strength of this cell wall, the cell membrane is going to be close to cell wall, but cell wall is not going to burst because of its tensile stretchy nature. So it's not going to burst. So this is the difference. If you put a plant cell inside distilled water, it's not going to burst in a lot of cases. As I have myself tested this. It expands. We have, I think it was an experiment during AS levels where we measured the length of a onion, which was placed in water before and after, and the length was increased, yeah? So the turgor pressure, the cell wall helps with the turgor pressure, and helps plant resist the turgor pressure, which results in bursting of the cell. Whereas because of animal cell not having cell membrane, they get bursted. Now, the mechanical support, I don't think I need to explain this, but plant cells have specific shapes because of cell wall. They provide mechanical support, Also, because they are like hard and stretchy structures, and there was some things about plasma dismeta which I've already explained. So this was enough for cell wall. The remaining part is going to be explained in the chapter of transport and in the animals. Oh, sorry, plants. Now, have I explained? No, I haven't. There is one thing more left, which is there are different small parts, and in plants there is a bigger part which is called a vacuole. So this is a vacuole. So in plants it's in tiny pieces and they are numerous in number, whereas in plants there's a large central vacuole. And vacuole, so vacuole is a fluid filled single membrane called tonoplast. Animal cells have relatively small vacuoles, and such vacuoles are called contractile vacuoles. Plant cells have large central vacuoles surrounded by a membrane called tonoplast, and the fluid they contain is cell sap. So they contain cell sap. I am not really writing a point here, but you should write it, that animals have numerous vacuoles inside them, and they are tiny and small, whereas in plants there's a large and central vacuole. Now, what does the cell sap contain? So cell sap is a concentrated solution of minerals, salts, sugars, organic acids, oxygen, carbon dioxide, pigments, some waste, and secondary products of metabolism. So it's like a storeroom where, again, I'm just repeating what is the composition of it. So it is a concentrated solution of minerals, mineral salts, which helps in muscle contraction too. We're going to be talking about that, but it consists of mineral salts, it consists of sugars, it consists of organic acid, it consists of oxygen, it consists of carbon dioxide and pigments and some waste products and secondary products of metabolism, you can say. So this was all about the cell wall. There's nothing really much about it, but remember one thing, that cell sap is usually more concentrated than the cytoplasm, as it is a storage area, so there is going to be more concentration of substances inside it. Anyways, let's just go towards the last organelle that we need to discuss here, which is lysosomes. So, what are lysosomes? So lysosomes are vacuoles. Yes, you heard me right. They're vacuoles which are, let me just drink some water here. So when we were talking about the vacuoles, the pathway of vacuoles, which was from the ribosomes, I'm just recalling it, ribosomes to rough endoplasmic reticulum. So this was a pathway of protein here, yeah? Rough endoplasmic reticulum, then Golgi apparatus or Golgi body, and then vacuole. So lysosome follows the same path because it contains proteins. It is a vacuole, but there's a question, but hold on. Why is it called a vacuole? Why isn't it called a vacuole? Why is there a different name for it? Because it has a specific function here. So the function of vacuole, lysosome. So what is happening to me? A vesicle. I take my words back. Vesicles. Vesicles. So lysosome is a kind of vesicle. Vesicles were, as I've told you, they are the single membrane structures that contain the proteins, the products that are formed from Golgi body, and they are transported outside intercellular and intracellular. So lysosome is a kind of vesicle which consists So, hydrolytic enzymes, as you can guess from the name, what does hydrolysis means? Because we have done the chapter called biochemistry or biological molecules, you know what hydrolysis means. And I'm waiting for you to recall it. Then I'm going to be telling you in a minute. So, hydrolysis is the process in which water is used to break down complex structure or molecules. Like for example, there are polypeptides, they are polysaccharides, and these are broken into monosaccharides, amino acids in the case of polypeptides. They're broken down into single units by the help of water, and the process that is involved in this is called the hydrolysis. So, hydrolytic enzymes are the ones that promote hydrolysis, the reaction of hydrolysis within the cells. Now you're going to think that, for example, this is an animal cell, and they're usually formed inside, or you can say they are inside the eukaryotic cell. So for example, this is a animal cell. I'm not going to build the whole diagram, but this is, for example, lysosome. So your next question is going to be, if it contains hydrolysis, that is going to be dangerous for the cell. Yeah, because if somehow the membrane breaks here, hydrolytic enzymes are going to be released inside the cell. It's going to be hydrolyzing the components of the cell, and this is going to kill the cell, destroy the cell. And this is basically what happens. It has an ability to do that. But what usually happens is, let me just give you an example here. Every country has an army, yeah, which is, or missiles, yeah. For example, Russia has a lot of different kinds of missiles, yeah. They have potential to destroy if somehow they, like Chernobyl, they somehow initiates themselves inside Russia. So there's gonna be damage to the Russians, yeah. So likewise, this is a missile, or you can say a weapon, that cell contains to destabilize, to kill bacteria, the things, or the things that are useless, or the things that can harm the cells. So these are basically the weapons here. This is the weapon technology that cells have. But in some cases, these weapons explode themselves, like the Chernobyl did. But Chernobyl was a mistake. But in the case of cells, they are done deliberately, and we're going to be talking about why do they do that. So, lysosomes are found in eukaryotic cell, found in eukaryotes. They are surrounded by single membrane and are simple sacs. They contain hydrolytic enzyme or digestive enzymes such as protease, nucleases, lipase, which breaks down proteins, nucleic acids, and lipids respectively. So let's just say simply a mosquito is here, hold on. Yeah, so they contain hydrolytic enzymes. I'll explain what hydrolytic enzymes means. Or you can say digestive enzyme. And I've named some of the enzymes too. Such enzymes carry out hydrolysis reactions and works best in acidic environment. Okay, usually, okay, so there is the composition of, there's the diameter of it. So the diameter of the lysosome is between 0.2 to 0.5 micrometers in diameter, but you can say preferably it's 0.5 micrometers. All lysosomes are vesicles, but not all vesicles are lysosomes. So this is a technical thing. Every vesicle cannot be called a lysosome. You know why? Because vesicles are the sacs which contains the products of the Golgi body, or you can say smooth endoplasmic reticulum, or you can say rough endoplasmic reticulum. So it can be other lipids and stuff. It can be anything which is the product that is produced inside rough endoplasmic reticulum, which is the processed protein. Smooth endoplasmic reticulum, which is can be the lipids and, for example, steroids and stuff. And they can also be the products of rough endoplasmic Golgi body, which are going to be conjugated molecules and stuff, or you can say highly processed or organized enzymes that are processed by Golgi apparatus. So all the vesicles are not lysosomes, but all the lysosomes are the vesicles. Yeah, they are formed from Golgi body, the pathway that I told you, and they are synthesized by Golgi body. Yeah? By following the pathway that, for example, ribosomes, rough endoplasmic reticulum, and then the Golgi body. They are responsible certainly for breakdown hydrolysis of unwanted structures such as old organelles, or even the whole cell which is invaded by a virus or other microorganisms. It can also digest mitochondria during cell starvation, because mitochondria can divide by binary fusion. Also, it contains a lot of amino acids, ATPs, and etc., which can be used by organisms for energy. So it can also be used for apoptosis, which is cell initiation of cell's own death or cell suicide. Yeah. So why does that happen? Let's just discuss that. But So it being used for digesting unwanted organelles, this is some basic things. So for example, if there's an organelle which needs to be disintegrated or be vanished from its existence, for example, let me just give you a scenario here. So this is the cell here, and these are, let me just redraw. So let's say, just for example, this is a cell here. Yeah. Now this cell is sad because it has not found any glucose since a long time, and it has not been provided. The person consisting of this cell does not eat, is in coma, let's say, and is not eating anything since a long time now. So now this cell is very sad and angry too, and it doesn't know what it should do. Yeah. So it goes into the medieval times when it doesn't contain, or it was not in symbiotic relationship with mitochondria. So what it does, it becomes greedy here, and it thinks that mitochondria looks delicious because it has ATP, proteins, nutrients, and a lot of things. And then it also thinks that although if I eat one, another one is going to divide into two, and because it just divides by binary fission, so it's not going to make much difference. Yeah. So during that time of starvation, when cell was angry, cell sends lysosomes to the mitochondria, which releases the hydrolytic enzymes and vanishes the mitochondria, or digests the mitochondria, providing the necessary nutrients for the cell's survival. So unwanted organelles. So in this case, mitochondria was not really was kind of unwanted, you can say, because there was no food coming, and when there's no food coming, there is no need of mitochondria, because mitochondria usually do the cell respiration, and when there's no food, respiration usually does not occur. So if there's no cellular respiration occurring, there is the need of food for both of the cell and mitochondria is then just a prey for the cell to be used. So this happens, and usually cell use these unwanted organelles, or you can say the organelles, during the time of starvation to be digested, and the nutrients used in it. Substances, for example, there is a substance which goes into the cell and it needs to be further broken down. So it is done by the help of lysosome. And even complete cells, for example, a virus can, this, for example, this, so this is a virus here, for example. It enters into the cell and goes inside the nucleus and takes control of the nucleus here, and makes cell do dirty things for it. So this specific virus has took control over the cell completely, because it usually goes inside the nucleus here. So because of its presence inside the nucleus, the lysosomes cannot enter nucleus. The immune system, which are lymphatic cells, B lymphocytes, they cannot enter the cell, obviously. So in this case, what is going to happen? What should we do? The virus has took control of all of the machinery inside the cell, and kind of what we can say is that the cell activities are controlled by the virus now. And what does virus do? Use this all machinery to produce copy of itself, which go and infect other cells. So what's gonna happen is, for example, this is a building which contains dangerous people here, and we know that they are here. We can't just throw rocks here, we can't just shoot firearms. So what we're gonna do is just throw a bomb here and annihilate the whole room. So this similar concept is used by the cells here. So because virus was inside and unreachable, what happens is some molecules, the messenger, chemical messengers, comes and attaches at the surface of the cell membrane, and we're going to discuss how it's done. But this, when a virus is detected by the body, it sends to the specific cell which contains the virus some signal molecules. What happens is that those signaling molecules activates apoptosis, which is cell initiated death, which is like, for example, the lysosomes inside the cell, which were to be used for hydrolyzing some substances and stuff, gets blasted, or the cell wall dissolves inside, and all of the lysosomes, so for example, I'm gonna represent lysosome with a rainbow color, so all of this lysosome just spreads inside the cell, killing the virus and killing the cell. So both of them are killed, and we're so sad. For killing one virus, we killed our cell. This is a sacrifice. This is a cell shield. This is a human shield that virus used. Anyways, let's just go back. So this is what happens, and this is how the cell-initiated death occurs. Anyways, so we're done with lysosomes. We're done. Okay, one point I should just mention here, which I've explained. So during starvation, cell digests its own mitochondria so that it can get nutrients, ATPs, amino acids which are essential for its survival. And also mitochondria. So they don't typically digest all the mitochondria, because nowadays the modern cell, the eukaryotic cell, cannot survive without mitochondria, so it is a necessity. But because mitochondria can divide itself via binary fission, so usually what happens is if we eat one, we have another made by one of the mitochondria reproducing itself. So it's not really usually a very big problem if the cell doesn't get the food after digesting mitochondria. Then it's gonna be dangerous, because there isn't gonna be a supply of glucose, or you can say energy, or ATPs, which are required for cell or its survival. So now, officially, this chapter is closed, but let me just mention here some of the things. So one of the points that I can mention here is that the newly formed lysosome, before it's starting its function, is called primary lysosome. Now, another organelle which is similar to peroxisome, because it is another kind of vesicle, so I should have just mentioned the types of vesicle, but let's say another type of vesicle is peroxisome. And it's mainly concerned with detoxifying alcohol. So, peroxisome is a kind of vesicle which contains enzymes that usually breaks down the toxic substances. For example, let's say alcohol. Another kind of substance which is hydrogen peroxide, which is found in cells at some places. It is also broken down by the enzymes present in the peroxisome. So the main function you need to remember here for peroxisome is the detoxification of some substances, or detoxification of specific certain substances which can toxify the cell, or which are harmful for cell. So it is usually concerned with the detoxification. And if you want to memorize, so just for an example, I gave you the example of alcohol, so just for a reference here, it is involved in detoxification of different substances too, which for example, I told you hydrogen peroxide, also alcohol, and other two, but you need to remember those two. Another kind of vesicle that we have here is glyoxysome, which contain enzymes that initiate the conversion of fatty acids into sugar. Or you can say, let's just say sugar. So glyoxysome is another kind of vesicle, which is obviously formed from plasma, or you can say Golgi body, using the pathway that I told you. And it also contains enzymes which are used in conversion of fatty acids into the sugar. So this one more thing I need to tell you is that the chloroplast is also named plastid. Mentioned here. This is another name of chloroplast here. And this is the end of our chapter. I think we have covered everything that was necessary for you to understand. And now, after this chapter, we are going to be starting on with the cell membranes. I'm sorry if I was stuttering a bit today. Looks like I'm a bit discombobulated because I haven't slept for, like, let me just think. Like yesterday I slept at 7 and I woke up at 11, so it's like 4 hours. So in the past 48 hours I've slept only 4 hours, and it's like I'm a bit stuttering right now, and I feel a bit dizzy, a bit, I can't even think right now, but the chapter is ended. I think there was one thing which is practically which is used in practicals. Let me just use the screen here. So there is one thing which I need to mention. I have though told you guys about them, which are the sizes of the cellular components. Like for example, mitochondria 1 to 3 micrometers, and for example, let's say chloroplast 3 to 5 micrometers, and for example, the cell which is 10 to 100 micrometers. So I've briefly told you the sizes of them, but I haven't written it down in the notes. So if I haven't wrote anything, I will provide you with those with a chart, you can say, which tells you the size of them. For example, the ribosomes were like 20 nanometers. So this is the thing that I'll provide you guys regarding the microscopic thing, which is used in practical to measure the sizes of cells. I think I should explain this in the next chapter after completing the cell membrane stuff. So when you're completed with the complete cellular descriptions, I'm going to be explaining that for a bit, because it's usually the part of paper 3, which is practical, and it's not very hard. You just need to memorize a formula, which is image size is equals to actual size multiplied with the multiplication or the magnification, you can say. So this one formula, and you need to know how to apply that. This is just it in that part. So about the processes that are used in the, you can say, knowing the chemical composition of cells, which is centrifugation, how the components were discovered by using centrifugation. What is centrifugation? It is not required in A levels. You don't need to study it. But for the reference here in the MCAT, I'm just going to say that there are different techniques that are used for knowing the cellular components and the cellular, you can say, determination of chemical composition, which is called cell fractionation. And usually what is used in those techniques are centrifugation, which is a powerful motor which spins the cellular components, and because of this, sedimentation occurs. So for example, if there is a larger by mass, there's a component, for example, let's say mitochondria, and ribosomes are in the centrifugation process. So what's going to happen is, on the pellet, which is on the bottom, the heaviest, you can say, component is going to be sedimented at the bottom, whereas the ribosomes are going to be spinning around it. So different components sediments at different speeds, and by that we can measure their mass, their size. We can estimate that which one has larger mass, which has a lower mass. We can also know the chemical composition by a technique which is chromatography, which is separating different components of mixture. You have probably learned about this in the chemistry class in 9th, so I don't think I need to I need you to explain that. The two different you can say procedures that are used, one is spectrophotometer, and another one is, you can say, microscopy, which is obviously you know about it, but spectrophotometer, and another one is the electrophoresis. Now, electrophoresis is a technique which we will discuss in detail in the chapter of genetics, because it usually involves in the separation of proteins. The heavier there's a gel and electricity is passed through it, and the molecules which are heavier moves and stays behind, and the ones that have higher charge density and are lighter moves faster. So this is a whole technique of electrophoresis, but it is usually involved in the you can say separation of different kinds of proteins here, or you can say nucleic acids, the charged molecules. For instance, the charged molecules are separated by this technique, and though cellular components are charged, the proteins are charged, so they can be you can say separated by this electrophoresis technique. We're going to be talking about this process. We're going to be talking about the what you can say what apparatus you need what is the technique, what are the things used in this technique, what happens in this technique, and what are the results, and everything is going to be discussed about this technique in, you can say, genetics. Whereas the spectrophotometry, it is a method of measuring light absorption by a particular substance. So for example, every different substance has a specific absorption spectrum of light. So for example, I'm just briefly explaining it because it's not involved in A levels. It might be involved, it might be asked in the MCAT, maybe, I am not sure about it, but I still need to explain these bits and tiny details that can or cannot come. So about the spectrophotometry thing, so for example, there is glucose and there is protein, yeah. So if you passes light through a molecule of protein and a molecule of glucose, both of them, because of different atomic configuration or different chemical compositions, the absorption spectrum, or you can say the light which is absorbed, the spectrum of light which is absorbed by both of those molecules is going to be different, and we can identify on the basis of that these two molecules are different. And because carbon has a different absorption spectrum, hydrogen has a different absorption spectrum, you can say the bond length, for example, CH bond has a specific absorption spectrum. The C bond O or OH or double bond O has different absorption spectrums. So if we passes a light through C bond with H between the bond, so the bond absorbs light of a specific spectrum. And if you passes a light through C double bond O, which is in for example, let's say ketones in the glucose, there is a different absorption there. So there is a graph that is going to be plotted, which is going to be telling all the absorption spectrum, the bonds involved in the absorption spectrum. Let's say glucose doesn't have nitrogen and hydrogen bond. So if there's going to be an absorption spectrum being built or being drawn, so during because by the light photospectrometer, or what was the name? Sorry, I missed the name. Spectrophotometry. So if we draw the graph of spectrophotometry here, there is going to be a peak at the nitrogen and hydrogen bond. You can say the wavelength of the absorption spectrum of nitrogen and hydrogen. So this is gonna tell us that nitrogen and hydrogen bonds are present in the molecule. Whereas if we pass the light through glucose, there is gonna be zero peak at the absorption spectrum level of the nitrogen and hydrogen, which is, I don't know how many nanometers of wavelength, but let's just assume that there is no peak at that absorption spectrum. So we're gonna deduce that because there is a nitrogen-hydrogen bond absorption at the molecule A and there is no at molecule B. So the molecule A, which has nitrogen and hydrogen bonds absorption, it might be protein. And the one with glucose, it might be glucose. So there are different kind of bond spectrum, the absorption spectrum of different bonds, which on the basis of which we determine which molecule is it. And you will learn about this in A-level chemistry in very detail for like in A-level, which is A2. So yeah, I think I have explained everything regarding these cells. I hope you liked the video. I hope you understood everything. If you have any questions, ask in the comments. Until then, bye-bye.